Hello everyone, today we will be talking about Kingdom Animalia Part 2. Now this is Part 2, so if you haven't watched Part 1, I encourage you to stop this video, go watch that first. In our last video we talked about different ways that we can characterize animals in Kingdom Animalia. Things like body symmetry, so are they asymmetric, bilateral, or radial? We talked about segmentation. Do they have segments in their body? We talked about the germ layers, so the ectoderm, the mesoderm, the endoderm. And we also talked about body cavities, so acoelomate, coelomate, and we briefly talked about pseudocoelomates. Now, you're not going to need to know all of that information for all of the phylum we're going to be talking about today, but it is a good idea to have an example for all of those. And like I said, we're going to be talking about the different groups of animals. We're going to be focusing on the invertebrates because those are the ones that you will be least familiar with, but we will also touch on the vertebrates. So we're going to start with the invertebrates. Now, invertebrate means they do not possess a dorsal nerve cord because a vertebrae is your backbone and they don't have a vertebrae. So they don't have that backbone with the dorsal nerve cord, so we call them invertebrates. As you may recall from our last lesson, these make up the majority of Kingdom Animalia, and yet they're often the ones that we are least familiar with. Take a look at these images down here. You probably recognize all of them, and yet you don't know what phylum they're in. So we're going to talk about that. By the end of this, you should be able to put all of these into their correct phylum. You may also be able to use that list. Can you pull up in the list from our first lesson? You may also be able to put your list of organisms from the first lesson into the various phylum that we're going to be talking about. So our first phylum is the phylum Porifera, or basically sponges. Okay, so our example organism is the sponge, and you can see our happy little sponge up here in the right-hand corner. Now, these are the most primitive group within Kingdom Animalia. They are sessile, and they have no true tissue. So when we're talking about those germ layers, Porifera don't have those germ layers. Now take a look at one of these images. Can you figure out what type of symmetry it has? Is it asymmetric, radial, or bilateral? If you need to, pause the video and go double check what those three types of symmetry are. Hopefully you paused the video, or maybe you just knew. Our sponges are our only group that are actually asymmetric, so you can't draw that line of symmetry anywhere along their body shape. Now, we say that Porifera are primitive because, well, for one thing, they don't have those true tissues, but they also, as a result of that, don't have any sort of systems. And what you see here is this central cavity, if we take a look at a sponge, and water is drawn out and pushed out. And this central cavity acts as sort of the stomach of the sponge. And these cells that line it are going to be working on absorbing and capturing any particulate matter that comes into that central cavity. So they're simple, but by no means are they bad at what they do. So those were the sponges, or the Porifera. Next up on our Kingdom Animalia phylum, we have our Cnidaria. Examples of Cnidaria that you're familiar with will be coral, sea anemones, and jellyfish. So all of these guys down here. Now, when we're looking at a model organism for phylum Cnidaria, you're most often going to be looking at jellyfish. And so here's a little jellyfish over here. And here we have a polyp, which is part of the life cycle of a jellyfish. And actually what happens is you get jellyfish budding off from the polyps to create, well, jellyfish. Now, while our sponges, or our Porifera, were asymmetric, our cnidaria are radially symmetric, which means it is symmetrical along any plane through the central axis. So if we draw a line through the top of it, it is going to be symmetric on either side. Similar to the sponges, they have a single opening. So the sponges, well, sort of had a single opening. The cnidaria also have a single opening, and you can see it right there. While sponges had no germ tissues, so no actual tissues, our cnidaria have two tissues. They're going to have the ectoderm and the endoderm, so the inside and outside, but no mesoderm, no middle layer. So that germ layer that creates the muscles and the bones, that's not going to exist in our happy little cnidaria. Now, because there is only one opening, that means that this creature has an incomplete digestive tract. It doesn't go all the way through the creature, so we say it's an incomplete digestive tract. Given that corals are part of this phylum, there's an obvious important ecological factor because our coral reefs are a source of food and habitat for many, many species. Now, one other interesting thing about our jellyfish or cnidaria is that they all contain stinging cells. These stinging cells are useful because it can help to capture prey or defend against predators. Now, not that it's an important detail, but I do see a bit of a typo on this slide. The cells, the stinging cells, are actually called cnidocytes, and within the cnidocytes, we have nematocysts, which are sort of the structure inside that allows the cell to be a stinging cell. And, it's not advisable for humans to touch jellyfish, and the reason is because they can sting us, and that hurts. So the jump from the Porifera to cnidaria included a few significant body plan changes, including symmetry change, as well as presence of actual tissues. Next up, we're going to talk about the platyhelminths. Now, the platyhelminths are known as flatworms, whoops-a-daisy, because they look kind of like a worm, but they're flat. If we zoom in, here's our little creature. This is a planaria. Planaria are utterly adorable. They have these little eye spots on their anterior portion of their body, and it looks like they're crossing their eyes, and it's adorable. So let's take a look at our planaria, our little flatworm. Just by looking at it, I know you only see a little bit, do you think that it's asymmetric, radially symmetric, or bilaterally symmetric? If you need to, pause the video, but I want you to try and figure that out. Now, hopefully you paused and were able to figure out that it is actually bilaterally symmetric because I can draw a line down its middle, separating the left side from the right side and creating that mirror image. And if I wanted to, I could go over to my other drawing and do the same thing. Oh, not very well though. And so these guys are different from the cnidaria, the jellyfish, because they are bilaterally symmetrical, which means you can draw that line down the middle and have a left side and a right side. But they're similar because they have an incomplete digestive tract still. So they have one opening. They have a mouth and an anus, and it's all in one spot. Now, because they're bilaterally symmetric, they're going to have that cluster of sensory organs on their anterior portion. So they're going to have like a head-ish region. And if you recall from our last lesson, that's one of the benefits of being bilaterally symmetric. You can sense in the direction that you're going in. Now, these flatworms, besides looking totally different, are also different from our cnidaria, our jellyfish, because they have all three germ layers, which means they have the ectoderm, the endoderm, and the mesoderm. So just to summarize some major differences is we now have bilateral symmetry, we now have three germ layers, and yet we still have an incomplete digestive tract. Next up on our phylum, we have another worm. So we just looked at flatworms. Here we're going to be looking at roundworms. Now these roundworms also have bilateral symmetry because we can draw a line down their midline. But they're different from our flatworms because they finally have a complete digestive tract, which means they have a mouth and an anus. So food comes in one space and exit as waste out another area. Now, where our flatworms, or platyhelminthes, were considered to be acoelomate, our roundworms are considered to be pseudocoelomates, which means they do have a cavity, but it's called a false cavity, so it's not quite the same as a true coelomate. These worms are still unsegmented, so there's no segmentation in their body along the way. But just like our flatworms, or planaria, or platyhelminths, they have 3 germ layers, and everything going forward is also going to have those 3 germ layers: ectoderm, endoderm, and mesoderm. Here is our last worm. So we've had 3 worms: the platyhelminths, the roundworms, the nematodes, and now we have the phylum Annelida. Phylum Annelida are our segmented worms. So they have segments which divide the body into repetitive sections. And if you recall, there's some advantages to this. So you can damage a single part, but other segments can still function. And we can also have segments that move independently of one another. Our classic example of an organism within phylum Annelida is earthworms. Those are worms you should be pretty familiar with. And leeches are another example of an organism in phylum Annelida. And these worms are finally coelomates. So we've had 3 germ layers for a while. These worms are the first that are going to have coeloms, which is those body cavities inside them. If you recall, there's some advantages to a coelom. It helps protect the organs within the organism, and also allows for more complex development of organ systems. And as a result of that, partially as a result, you're going to finally see what is known as a closed circulatory system. This is a topic that we've explored. Some of us, you may have explored it while we were talking about circulatory systems, types of circulatory systems. And if not, here's a quick refresher. So an open circulatory system is basically when blood sloshes around the cells. And this open circulatory system is often considered to be less efficient because you can't control where the blood is going. Meanwhile, organisms in kingdom Annelida will have a closed circulatory system, which means blood is contained in vessels. So, for example, humans have arteries, veins, and capillaries. And those allow the blood to be directed in certain areas of the body. So our worms have a closed circulatory system, and they also have a complete digestive system. So they have a mouth and an anus. And these are our first guys in the animal kingdom to be segmented. Here's another phylum you will know very well. This phylum is Arthropoda. Arthropoda includes insects, crustaceans, and arachnids. So insects, spiders, and many water creatures like lobster or shrimp. All of these creatures have this hard exoskeleton. So they don't have a skeleton like humans. They have what's known as an exoskeleton on their external body. So it's on the outside of their body. And it is made up of chitin. Can you remember where you've heard the word chitin before? If you need to, pause the video, give it a bit of a think. But chitin is also a chemical found in the cell walls of fungi. So just like our earthworms or our phylum Annelida, arthropods are also segmented. At this point in time, the segments are a little bit harder to visualize, but they may be things that you're familiar with. They have a head, a thorax, and an abdomen. And out of these body segments, you're going to have appendages and different specialties of these things. So for example, the head might have many of the sensory organs like antenna, whereas the thorax and abdomen might have different functions. Just like many of the creatures we've already talked about, they have that complete digestive system, so they have a mouth and an anus. But unlike our annelids, they actually have an open circulatory system, so that the circulatory fluid, which in humans is blood, is going to slosh around their tissues. This phylum is the biggest animal phylum. It has so many species in it. And the species are also very diverse. Interestingly enough, it is the only phylum that has invertebrates that can fly. I want you to pause the video and consider what kind of ecological role the organisms in phylum Arthropoda have on ecosystems, and what kind of impact do you think that they might have on humans? Pause the video. Hopefully, you took a minute to pause it. I'm going to go on. So they're definitely important in ecosystems because they're usually going to be primary consumers, which means they're that first step, or they're the first things that are going to be consuming the plant matter. They're also really important for as pollinators. They're going to be carrying pollen from flower to flower, allowing for sexual reproduction of plants. Now, in terms of how they impact humans, they destroy crop. Thousands and millions of dollars of crop is destroyed basically by insects. And so in a way, we compete with these insects for resources. So they're very problematic, and part of the reason why we use things like insecticide. Here's another phylum you should be familiar with. It is phylum Mollusca. So the mollusk phylum includes things that usually have a shell or some sort of modified shell. So things like snails, mussels, clams, oysters, and also squids and octopi. Now, obviously squids and octopi don't have a shell, but squids actually have something called a pen, which is a modified shell that's internal to their body. And octopi have no shell at all, and for both of them it's believed that they once had a shell, and they have evolved to no longer have it because they were able to move faster and compete better against their shelled relatives. So just like the arthropods, our mollusks are going to have an open circulatory system, but there are exceptions like the cephalopods, which include things like squids and octopi. So those will be our snails, mussels, clams, and oysters will have an open circulatory system. But like I said, there's exceptions, and that's okay. They do still have that complete digestive system, which means they have a mouth and an anus. And same as our organisms in the past, they're still bilaterally symmetric. The one difference than our arthropods is that they're not usually considered to be segmented. And just like our annelids, so our earthworms, and our arthropods, these mollusks are going to be coelomates, so they have that body cavity inside them. And finally, our last invertebrate phylum that we're going to talk about is the echinoderms, or echinodermata. The organisms in this phylum are the ones that you're going to be most familiar with are our sea stars, our happy little sea stars. There's also urchins and sea cucumbers within this phylum. Now, these echinoderms break from the tradition a little bit because they go back to being radially symmetric. Interestingly enough, though, their larval stages, so a different stage of life than their adults, are bilaterally symmetric. So we are going to consider them to be radially symmetric, but just know that they do have some stages of life that aren't radially symmetric. These organisms, so our stars, urchins, and cucumbers, have a complete digestive system, but an open circulatory system. So their circulatory fluid is going to be sloshing around, just like in the arthropods and the mollusks. Now, one thing that's really cool about echinoderms is that they actually have a water-filled vascular system that uses hydraulics to pump water around their body, and they can control the movements of their arms and feet, well, their tube feet, using hydraulics, which I think is just really cool. So that is it for our invertebrates. We talked about our sponges, the porifera. We talked about the cnidaria, the jellyfish. We talked about the platyhelminths, the flatworms. We did not talk about the rotifers. We talked about phylum Mollusca, which are our shelled creatures like snails, clams, but also octopi and squid. We talked about our annelids, which are our segmented worms, so for example, leeches or earthworms. We talked about nematodes, which are the roundworms. And we talked about arthropods, our insects. And we talked about echinoderms. And so some of these may be familiar to you, and some of them may be brand new. And with each of these, we talked about some differences in their body plans, whether it's symmetry, asymmetric, bilateral, radial, whether it was number of germ layers, whether it was presence of segmentation, presence of a coelom, and we also talked about whether they have an open circulatory system or closed, and if they have a complete digestive tract, so mouth and anus, or just one opening, which is a incomplete digestive tract. So those were the invertebrates. Now we finally get to talk about the vertebrates. We are not going to go into each of their phyla because you are pretty familiar with them. There's the fish, amphibians, birds, mammals, and reptiles, all organisms that you're familiar with. And if I were to hazard a guess, that initial list that you created on our first lesson would have included organisms primarily from these groups of under kingdom Animalia. Now, one thing that's also kind of interesting about the fish is we have a few different types. We have what is known as the jawless fish, so our lamprey over here has no jawbone. We also have our cartilaginous fish, so sharks are that under fish, but also they don't have any true bones. And then we have our bony fish, so things like tuna. And so our chordates have some common characteristics that I want to talk about. The main characteristic of the chordates is that they have a vertebrae, or they have a backbone that supports a nerve cord. That's what makes them under phylum Chordata. They also have we also have an internal skeleton. So we have our bones, and these bones can grow with us as we age. This is very different from the arthropods who have that exoskeleton, so that skeleton on the outside, because anytime the arthropods get too big, they have to shed their exoskeleton in a process called molting. As a result of this internal skeleton, it helps us move in more complex ways, and we also have a skull that contains and protects our brain, and our vertebrate actually extends past the anus to a tail. All chordates are going to have some form of gills or lungs or a mixture between the two. We are all going to have a ventral heart as opposed to dorsal. And we all have a closed circulatory system as opposed to open. So closed meaning it's the fluid is going to be contained within vessels. And this is different than our mollusks or our arthropods. As far as some other general characteristics, we have 2 pairs of appendages attached to our torso. So we have 2 arms and 2 legs. We also have a multi-layered skin. So it's not just one layer of skin cells, it's several layers deep. And we have accessory structures that help us do various functions, whether it's hair, feathers, glands, claws, horns, hooves, nails. And these accessory structures help us chordates to be better at whatever it is we do. Now, I know that I said invertebrates take up the most number of species in kingdom Animalia, and they're very diverse. But vertebrates also have a lot of diversity. We are diverse in the sense of size. There is a tiny, tiny hummingbird that only weighs about 2 grams. And then you can compare it to blue whales that have a mass of, I don't know, they're huge. They're huge. We also have a huge range of sensory abilities. Now, humans, of course, can see, touch, feel, whatever. But some organisms can do amazing things, like bats can echolocate, or pit viper snakes can detect infrared radiation. Our behavior is super varied, from migration routes to hibernation to how we respond to our environment. And then, of course, life cycles. There are many, many different life cycles that exist in the chordates. If you take a look at a phylogenetic tree for the chordates, you're going to recognize a few names on here, like mammals, aves, which is birds, reptiles, amphibians, and there's a few here that you won't recognize, but basically those are our fish. And over time, because remember, to read a phylogenetic tree, up here is present day, and back here is past. So regardless of how much time has passed, over time, we've had changes occur, and those changes are indicated here. So for example, at some point in time, organisms in phylum Chordata developed a bony skeleton, and that allowed them to later develop limbs and so on and so forth. And so if we go back to the whole picture again, here's our invertebrates over here, and our vertebrate over here, shown by a fish, what looks like probably a bony fish. And it all comes back to our ancestor, that colonial flagellate protist, which allowed all of kingdom Animalia to come to be what it is as we know today. So today we focused on the different phylum in kingdom Animalia, with a particular focus to the invertebrates, because those are the ones you would be the least familiar with. And within each phylum, we discussed some of the ways that they meet those classification systems. So their body symmetry. Are they radial, bilateral, asymmetric? It's good because if you know of an example of the phylum, then you can often figure it out by just picturing the example. Whether or not they have a body cavity. The earlier ones generally do not have a body cavity. The later ones often do. We talked about their germ layers. Sponges have no tissues, so no germ layers. Cnidaria have 2. Everything else has 3. And we also talked about segmentation, which jumps around a little, as does their open or closed circulatory system. Now, we talked about a lot of details for a lot of phyla, and don't feel like you need to know all of them. It would be good to know the common name or an example organism for each of the phyla, as well as a few basic characteristics for them. What's really amazing is how diverse each and every phyla is, and how good they are at what they do. Whether they're simple organisms or complex. The power of evolution and the beauty of diversity that exists in Kingdom Animalia. But that's it for now. As always, please let me know if you have any questions, and good luck.